Master the core mechanics of the gastrointestinal system with a dive into the regulation of salivary and gastric secretions. From neural triggers to hormonal pathways, we have the second part at the pathologist canvas by Dr. Nikki Susan Thomas.
Hello everyone. So let's continue with the saliva and salivary secretion. So we were about to discuss about the regulation of salivary secretion. Salivary secretion is regulated exclusively by the nervous system via the autonomic nervous system. Alright. So the parasympathetic fibers as well as sympathetic fibers are involved in the autonomic system. So autonomic nervous system has got two phases, that is the parasympathetic as well as the sympathetic nervous system. So under parasympathetic nervous system or parasympathetic fibers, first of all, we know that there are three types of salivary glands, major salivary glands, that is the parotid, the submandibular, and the sublingual. So the parasympathetic supply to each of it is different. So first of all, let us take submandibular and sublingual. So the preganglionic fibers arise from the superior salivatory nucleus. This is the nucleus that is present in the pons, and it travels via the nervus intermedius, the geniculate ganglion, then the chorda tympani, which is a branch of the facial nerve, the lingual branch of the trigeminal nerve, and then it goes to the submandibular ganglion. And the post-ganglion, so they have reached the ganglion, and now the fibers that comes out from the submandibular ganglion is known as the postganglionic fibers. So the postganglionic fibers will supply the submaxillary, that means the submandibular and the sublingual salivary glands. So this is the regulation of salivary secretion when it comes to the submandibular and sublingual salivary glands. Now we have the most important, that is parotid. Again, this is the parasympathetic fibers. All right. So in case of parotid, the preganglionic fibers arise from the inferior salivatory nucleus. In case of submandibular and sublingual, I told you it is from the superior salivatory nucleus, which is a nucleus of the facial nerve. But here, the inferior salivatory nucleus is a nucleus of the glossopharyngeal nerve. So the preganglionic fibers arises from the inferior salivatory nucleus, which is in the upper medulla, and then it travels via the tympanic branch. Tympanic branch is a branch of the glossopharyngeal nerve. So it travels through the tympanic branch of glossopharyngeal nerve, then the tympanic plexus, then it goes to the lesser petrosal nerve, and then finally to the ganglion, and that ganglion is known as the otic ganglion. And then what happens? The postganglionic fibers reaches the parotid via the auriculotemporal nerve. So the ganglion here is the otic ganglion, and the postganglionic fibers from the otic ganglion travels via the auriculotemporal nerve to the parotid gland. So you can draw this as a flowchart for your exam. And then let's see what is the effect that is shown. So the stimulation will produce large volumes of watery saliva. I already told you the parasympathetic system, its stimulation will lead to the secretion of large volumes of watery saliva, and it has got low organic content. Low organic content, and it activates the acinar cells by dilating. So here dilation is happening. Dilation of the blood vessels, because the neurotransmitter here is acetylcholine. This is the basics of the parasympathetic supply to the superior to the submandibular, sublingual, as well as parotid. So write it separately. That is, the parotid should be written separately because it has got the inferior salivatory nucleus, and then the branches are the tympanic branches, the tympanic plexus, then lesser petrosal nerve, then it goes to the otic ganglion, and via the auriculotemporal nerve, it goes to the parotid gland. And when it comes to the submandibular and sublingual, the impulses arise from the superior salivatory nucleus, which is within the pons. Then it travels via the nervus intermedius. So understand that this entire thing is guided by the facial nerve in case of submandibular and sublingual. And in case of parotid gland, it is by the glossopharyngeal nerve, which is the ninth cranial nerve. Fine. And then just continuing with the submandibular sublingual, then what happens? The superior salivatory nucleus, and then nervus intermedius, geniculate ganglion, then chorda tympani, and then it reaches the uh, it reaches a branch of the lingual branch of the trigeminal nerve, that is the fifth nerve, and then the submandibular ganglion. All right, and then from there the postganglionic fibers will directly supply the submandibular and sublingual salivary glands. But here it was via the auriculotemporal nerve that the entire impulses travel to the parotid gland. Okay, and we saw the effects also. And when it comes to the sympathetic fibers, the preganglionic fibers arise from the lateral horns of T1 to T2 spinal cord segments, and they synapse in the superior cervical ganglion. Superior cervical ganglion. And the postganglionic fibers travel along the arterial plexus. Arterial plexus to the glands, that is to the submandibular, sublingual, as well as the parotid. So the stimulation of the sympathetic nervous system will lead to the production of thick, mucus-rich saliva. In case of parasympathetic, I told you that it was more of watery saliva, but here we have thick, mucus-rich saliva because here vasoconstriction is happening, or vasoconstriction of the blood vessels is happening, that leads to production of a thick mucus-rich saliva. And here, which is the neurotransmitter present? It is the noradrenaline. Okay. And this parasympathetic as well as the sympathetic nervous system that regulates the salivary gland is guarded by certain reflexes. Okay, you know what reflexes mean, right? So these reflexes, there are several types of reflexes. So the two types of reflexes, that is controlling the sympathetic as well as the parasympathetic nervous system here, is the unconditioned reflex as well as the conditioned reflex. So from the word unconditioned, it means that it is not conditioned prior. So it is an inborn reflex which is present since birth that has got no prior experience. That is the important term here. There is no prior experience in case of unconditioned reflex. So it is triggered by any substance that is placed in the mouth. For example, in case of dentists, we know that when we take the impression of a patient using alginate, there is a sudden gag reflex that occurs and there is drooling of saliva in the mouth. So it makes our experience as well as the patient's experience very unpleasant, right? So that is an example of an unconditioned reflex because it has, the patient might not be experienced, the patient might not have taken an impression earlier, so it was for the first time. Okay, so this is an example of unconditioned reflex. Or when you place food in the mouth, that is also, for example, when you're trying a new dish for the first time. So that is an example of an unconditioned reflex. Whereas conditioned reflex requires prior experience. So there is a prior experience. That means it does not require any food in the mouth.
it is triggered by just the sight of food, the smell of food, or the sound. That means when even the thought of food can lead to salivation in the mouth. Okay, so the impulses from the eyes, nose, or ear will trigger salivary secretion. So that is conditioned reflex. So these sympathetic as well as parasympathetic nervous system is controlled by these two types of reflexes. And then the secretion of salivary gland also depends upon certain substances. So the substances that increases as well as decreases the salivary secretion is taken to be care of. So what are the substances that increases the salivary secretion? We have drugs called sympathomimetics. Sympathomimetics. So they are adrenaline and ephedrine. So they can increase the salivary secretion. Then we have parasympathomimetics, and examples of these are acetylcholine, pilocarpine, muscarine, physostigmine, and another substance that increases salivary secretion is histamine. And what are the substances that reduce the salivary secretion? They are the sympathetic depressants like ergotamine and dibenamine. Then we have parasympathetic depressants, which are substances like atropine and scopolamine. Okay. So these are the drugs that increases salivary secretion as well as decreases the salivary secretion. And we should also know about certain important clinical scenarios or applied physiology where saliva plays a role. So there can be both hyposalivation as well as hypersalivation. Hyposalivation means salivation is reduced due to various conditions, and hyposalivation means the salivation is increased. So hyposalivation can be due to hyposalivation. Hyposalivation can be due to reasons like fear, fever, so even anxiety, okay, fever, or most importantly dehydration, which is the most important cause. And there are certain permanent conditions that leads to hyposalivation, like the salivary duct obstruction. So that is known as silolithiasis. Lithiasis means if there is a stone in the salivary duct, that leads to the obstruction of the salivation. Or there can be certain congenital conditions, congenital reasons. That means the conditions that arise since birth, which can be in the form of hypoplasia of the glands, then some other conditions like Bell's palsy, and all these can lead to the most important consequence that occurs because of hyposalivation is dental caries. And another condition is hypersalivation. So this means there is excess saliva secretion, and one physiological cause is pregnancy, and the pathological cause is known as chiasm or sialorrhea. And some other reasons like decay, tooth decay, then gastrointestinal diseases, neurological diseases like Parkinsonism, cerebral stroke, psychiatric conditions, nausea. When we are nauseated, there is hypersalivation, right? And another clinical condition is xerostomia. Xerostomia is the word for dry mouth. So the causes are definitely dehydration, renal failure, or a syndrome known as Sjogren's syndrome, which is the most important syndrome associated with the xerostomia condition. Then in case of patients who have undergone radiotherapy for cancer treatment, then trauma, then various side effects of drugs that which we already saw, drugs like antihistamines, antidepressants, okay, and that this xerostomia can result in difficulty in swallowing, and also we know if there is no saliva, there is difficulty in speech, and it can also lead to halitosis or bad breath. Okay, then some other physiological some other conditions associated with saliva is, another one is drooling, that we already know what drooling is. It is known as stylism or excess salivation. And another one is cauda tympani syndrome. Cauda tympani syndrome. So this is a syndrome wherein there is sweating while eating, which is due to regeneration of any damaged parasympathetic nerve fibers which joins with the sweat glands after any surgical or a traumatic injury that occurs. Okay, so what happens when we eat? There is sweating. And another viral condition is mumps. You already know what mumps is. It is an acute viral infection which is caused by the paramyxovirus. So there will be puffy cheeks, fever, sore throat, etc. And also there will be problem with saliva secretion. And Sjogren's syndrome we already discussed, which is one of the most important syndromes associated with xerostomia or dry mouth. Alright, so that's about the saliva secretion, the regulation of salivary secretion, as well as the applied aspects related to salivary secretion. Okay. So up next, we're gonna have our discussion over gastric secretion. Gastric secretion. So we've already discussed about the functional anatomy of the stomach. I told you that the stomach is a hollow organ that is situated below the diaphragm on the left side of the abdominal cavity, and its empty volume is 50 milliliter, and its normal capacity is around 1 to 1.5 liters. So this is the normal capacity, but it has also got the capacity to expand up to around 4 liters. Okay, and let's see the parts of the stomach. So the stomach has got the first part, the first region, that is the cardiac region. Okay, and then the fundic region, and then the body, and finally the pyloric region. These are the parts of the stomach. So the cardiac region is the upper part. Okay, this is the cardiac region. So this is the region where the esophagus opens, and it is guarded by the cardiac sphincter. So there is a cardiac sphincter here. It opens towards the stomach. And this is the fundus. Fundus is a small dome-shaped structure that is elevated above the esophageal opening. And then comes the body. The body is also known as the corpus. So the body is the largest part. That is, it constitutes around 75 to approximately 80% of the stomach, and it extends from just below the fundic region to the pyloric region. And the pyloric region is the lowermost region. It has got andrum as well as a, this is the andrum. Okay, it has got andrum as well as a pyloric canal is also present. Okay, and it is separated from the body by a structure that is known as incisura angularis. Okay, separated from the body by the incisura, I'll write it here, incisura angularis. And then the pyloric canal opens into the duodenum via the pyloric sphincter. So there is a pyloric sphincter here. So that is the anatomy. So what are the glands of the stomach? So the stomach has got several important glands. These are the gastric glands. So the first one is the fundic glands. So the fundic glands are present at the body and the fundus region. Okay, body and fundus, body and fundic region. Okay, so from the name itself, that is self-explanatory. And then
It is also called as the main gastric or the oxyntic glands. Okay, funding glands are also known as the oxyntic glands, and the key cells which are present here are the chief cells, parietal cells, mucous cells. Then there is another group of cells called the EC cells, that is enterochromaffin cells, and we also have enterochromaffin like cells, that is ECL cells. So these are the major cells which are present at the fundic glands. Okay, these are the key cells that comprises the fundic glands. And then we have another important gland that is known as the pyloric gland. Pyloric gland. So the pyloric gland produces, it is usually located at the pyloric region, and the key cells present here are the G cells that secretes gastrin. We'll learn about gastrin later on. Then mucus cells are present, EC cells as well as ECL cells. Okay, so that's about pyloric glands. Sorry, that's about yeah, pyloric glands. And then we have the cardiac glands. The cardiac glands are located at the cardiac region. So just look at this image. It is located at the cardiac region. And since it is located here, it is short and it will be tortuous. And the main cells present here are the mucous cells, the EC cells, endochromaffin cells, and the endochromaffin like cells, as well as the chief cells are also present. So you have to study this thoroughly, otherwise. Now we're going to next move on to the secretions of each of the cells. So please study in particular you have to study about the types of glands present, the gastric glands, that is fundic glands, the pyloric glands, as well as the cardiac glands. And out of this, the fundic glands produces, or you cannot say it produces, the main cells of the fundic glands are the chief cells, the parietal, mucus, EC cells, ECL cells. Pyloric gland produces the G cells, mucus, EC, and ECL cells. Cardiac glands produces again mucus, EC, ECL, and chief cells. Okay, so that's about the glands. And now we are going to see the secretions of each of these cells that belongs to various glands. Okay. Okay. So first one is the cell that we're going to discuss here is chief cells. Chief cells, as I told you, you can see chief cells here. You can see that it is produced both at the fundic glands and also at the cardiac glands, right? And one more thing I forgot to mention is that these fundic glands has got a when you compare the total structure, okay, so this pyloric and cardiac glands have got more short and tortuous structure, okay, whereas the fundic glands, it is more elongated because its location is at the body and the fundus, whereas pyloric glands is located at the pyloric region because, and it's very short and tortuous, and the cardiac gland also is located at one end, so it is also short and tortuous. Okay, that also you need to know. So the chief cells produce, and one more thing, and one more addition, the chief cells are known as the pepsinogen cells. So from the word itself, we understand that the chief cell produces pepsinogen. Pepsinogen. And then another important secretory products by the chief cells are renin, then lipase, gelatinase, as well as urease. These are the main important enzymes which are secreted, or not enzymes, main secretory products produced by the chief cells. Then we have the parietal cells. Actually, the word parietal cell or the parietal cells have got another another name that is known as the oxyntic cells. Okay, so the parietal cells are also known as oxyntic cells, and they produce HCl, that is hydrochloric acid of the stomach, as well as the intrinsic factor of Castle. Okay, these are produced by the parietal cells. And then we have got the mucus cells or the mucus neck cells. The mucus neck cells produces mucin. Then we have the G cells. I told you the G cells produce gastrin, and there are EC cells as well as ECL cells, that is enterochromaffin cells, which produces serotonin, and ECL cells, which are endochromaffin like cells that produces histamine. Okay, so I'm just summarizing things from the textbook. So you need to read the textbook thoroughly after seeing this class. That makes it more easier for you. All right. Because once you read the textbook first, I think it'll be very difficult for you to understand because it's given in big, big paragraphs. So I'm making it much more easier for you to follow. Now, so these are the most important cells and their secretory products. Now let's see what are the various functions of the stomach. Functions. Let's use another color. Okay. So what are the functions? It has got mechanical. For exam, always try to draw flowcharts or diagrams and then explain it. Okay, so it has got mechanical, digestive, protective. Then it has got hemopoietic, excretory functions. These are the most important functions. Okay. So coming to mechanical functions, we know that the stomach has a storage capacity of around 1.5 liters. So it stores food for around 3 to 4 hours, maximum of 1.5 liters, and it will slow down the emptying. It allows proper digestion, and it also helps in chyme formation. Okay, so that helps in the peristalsis or the movement of this entire bolus of food with gastric juice into the semi-solid chyme. So that is the mechanical function. Now, talking about the digestive function, it acts via the gastric juice enzymes. We will see about the gastric juice enzymes in the next session. And then protective function is it is acting via the mucus in the gastric juice, because we know that acid, that is hydrochloric acid in the stomach, is highly acidic. It can erode the gastric surface, but since there is a protective layer of mucosa or mucous membrane, it is protecting the stomach from the erosive action of hydrochloric acid. And then the another function is hemopoietic, that is in blood related, that is it acts via the intrinsic factor of Castle. That also we'll see the explanation how it acts via the intrinsic factor of Castle. And then it has got excretory function because it excretes the toxins, alkaloids, and various metals are secreted via the stomach. So let's have a quick discussion on the properties of properties and composition of gastric juice. Okay. Okay. So this is the composition of gastric juice. We'll have a quick discussion on the properties first. So the volume of gastric juice is around 1200 to 1500 milliliters per day. It has got a specific gravity of 1.002 to 1.004. The pH is highly acidic. Why? Because of the hydrochloric acid. So it is around 0.9 to 1.2. And the composition is discussed here. Okay. Composition is made up of water, 99.5 percentage, and 0.5 percentage is solids. And out of the solids, we have organic substances as well as inorganic substances. And the organic substances are enzymes and other organic substances. Enzymes are pepsin, renin. It's RENNIN. Okay, that's a spelling mistake given there. Then gastric lipase, gelatinase, and urease. And then the other organic substances are mucus and intrinsic factor of Castle. Then the inorganic substances are the various elements like sodium, calcium, potassium, bicarbonate, phosphate, sulfate, and the acid that is hydrochloric acid. And the concentration of HCl is up to 150 milliequimoles per liter. So let's see what are the enzymes that is present or released by stomach. So first one is pepsin. Okay, pepsin is activated by hydrochloric acid of the stomach. Okay, so, so these are the digestive enzymes of gastric juice. We have already discussed about the secretions of saliva. So, and we have also already discussed about the properties of saliva. So, we had similar headings, but the values are different. So, please go through that again if you have forgotten that. And also, I have told you that for both gastric juice as well as for saliva and also for pancreatic secretions, we can follow a similar flow chart or a similar illustration of this kind. But the inside the box, it will definitely vary, but almost it is similar. So you can follow that, right? Around 99.5% will be water and 0.5% will be solid. So all that, I hope you remember what I've told in the first session. So coming back to the digestive enzymes of the gastric juice, first we have pepsin. So pepsin acts on proteins and the end products are proteoses, peptones and polypeptides. So pepsin will break down proteins into proteoses, peptones and polypeptides in the presence of hydrochloric acid. Gastric lipase. Lipase means lipid splitting or lipid breaking. ACE means it is splitting, right? So it also acts under acidic medium and it acts on the triglycerides of butter, and the end products are fatty acids and glycerols. Gastric amylase, again, it acts only at acidic medium. It acts on starch in the food, and then the starch is split into dextrin and maltose. Then we have gelatinase, which again acts under acidic medium, and it acts on gelatin and the collagen of meat. Our body, everything is made up of collagen. So it acts on the collagen, and then the end products are peptides. And next is urease. So urease is activated by acidic medium and it acts on urea, and the end product is ammonia, which is released from the body. Okay, and there is also another enzyme that is present, that is renin. Okay, renin again acts under acidic medium and it acts on casein, which is present in milk. Okay, casein of milk, and it will curdle this milk. So what happens? The end product will be curdled milk. So milk becomes curd here because of this. Okay, so these are the various functions of the digestive enzymes present in the stomach. Okay, so that was one of the functions that we learned, digestive function. And protective, I already told you, it is because of the mucous membrane. Now, what about hemopoietic function? Hemopoietic function is because the intrinsic factor of Castle is something that is secreted by the intrinsic factor of Castle. I'll write it as IFC. So, this is produced by the parietal cells, which is also known as the oxyntic cells that produces HCl. So these parietal cells release intrinsic factor of Castle, which is essential or very important for the absorption of vitamin B12. This vitamin B12 is known as the extrinsic factor. Okay, so please make this thorough because you will keep on hearing this in subsequent lessons. So the intrinsic factor of Castle is something that is produced by the parietal cells, and this helps in the absorption of vitamin B12, and this vitamin B12 is known as the extrinsic factor. So vitamin B12 is a key maturation factor during erythropoiesis or red blood cell formation. So that is why we say that it has got hemopoietic action. Right? So the deficiency of this will lead to what? Absence of intrinsic factor of Castle, and this can lead to a particular anemia, and that anemia is known as pernicious anemia. Soon after this GIT, I'm planning to take blood as our next topic of discussion. And there we learn about various types of anemia and various other disorders of blood. Okay. So there we'll discuss about this in detail. So these are the various enzymes and various actions of the stomach. And I hope you already know the functions of hydrochloric acid. First of all, it has got a very protective and bacteriolytic action because it kills bacteria entering the food. And one more thing is it activates pepsinogen into pepsin, because the enzyme that is shown here is pepsin, but it was first of all it was present as pepsinogen initially, and then it is getting converted to pepsin because of this hydrochloric acid. And this hydrochloric acid also provides acid medium required for hormone and enzyme actions. Okay. Next one is we'll have a quick discussion on the secretion of the gastric juice. Secretion of gastric juice, and we'll also have a discussion on the regulation of gastric juice. Okay. So the first of all, we are discussing in detail about the secretion of gastric juice as such. But we know that the gastric juice is made up of several enzymes and several factors and also HCl, right? So we have to take two things separately. We have to study about the secretion of pepsinogen separately and hydrochloric acid secretion separately. This is a very important topic. The secretion of HCl is a very important topic. I'll take that in detail. So, how is pepsinogen released? Pepsinogen is synthesized from amino acids in ribosomes of chief cells. Okay, the chief cells release, or the chief cells will have ribosomes. So the pepsinogen is synthesized from amino acids in ribosomes of chief cells, and they are packaged into zymogen granules. Zymogen granules. And then what happens? This is packed into zymogen granules, sorry, zymogen granules by the Golgi apparatus, and it is secreted into the stomach lumen and activated. So it is secreted to the stomach lumen, and then so when it is secreted, it is in the form of pepsinogen, which is inactive form of pepsin, and this is converted to pepsin by the action of hydrochloric acid. So that is how pepsinogen is released, or pepsinogen is secreted. And I told you the secretion of hydrochloric acid is really important. And please understand this diagram properly, because in case of hydrochloric acid, what happens is the secretion occurs in the parietal cell canaliculi. Parietal cells. So parietal cell is also known as the oxyntic cells. We know that the parietal cell produces HCl as well as the intrinsic factor of Castle, which has got a hemopoietic action. So the production of HCl occurs in the parietal cell canaliculi. So here carbon dioxide comes from metabolism or from blood. Okay, and then it combines with water. So this is inside the parietal cell. I think you can see this clearly. This is the parietal cell, right? It's shown here. So carbon dioxide enters the parietal cell either from blood or by metabolism, and it combines with water to form carbonic acid. Okay, so this is carbonic acid via the carbonic anhydrase enzyme. So this is the enzyme and this is the end product. H2CO3 is carbonic acid. All right. And then what happens is this carbonic acid splits into bicarbonate and H plus. Okay, so H plus is actively pumped into the canaliculus. So there is a canaliculus outside. So this H plus is pumped outside the canaliculus. So can you appreciate the way in which this particular entire thing is going? See, from here, it is going like this. It is coming here and then it is again going out, and then it is again coming in and then going out. So this is the pathway. So just learn the pathway. Okay. So carbon dioxide enters the parietal cell, combines with water to form carbonic acid with the help of carbonic anhydrase enzyme, and then this carbonic anhydrase splits into bicarbonate and H plus ions, and H plus ion is pumped into the canaliculus, okay, actively pumped, and then there is chloride from sodium chloride in the blood. Okay, so sodium chloride is present here, NaCl. So that will release chlorine. And this chlorine, what happens is chlorine from the sodium chloride in blood is pumped inside the parietal cell. So H plus goes outside the parietal cell and chloride or chlorine comes inside the parietal cell. Okay, it is pumped in from the sodium chloride in blood. And then what happens? This H plus will join with Cl minus, right? And then forms the hydrochloric acid. So this is how, and then here what happens? H plus, we can see here, right? H plus combines with Cl to form HCl. And then again, this entire process is happening again as a cycle. The Cl goes inside, then it goes outside. There also sodium is present, and then it forms sodium bicarbonate, and then bicarbonate is splitting here, it is going inside. So just learn it as a quick cycle. Okay. Now, okay. This is the most important part of the session. However, it is a little bit confusing also. But before studying the regulation of gastric juice or regulation of gastric secretion, we need to learn about three experimental methods. Experimental methods which were shown by scientists. So first one is the Pavlov pouch. The Pavlov pouch is a small bag which is incompletely separated from the stomach, and it retains the vagal as well as the sympathetic supply. Okay, so this, it is just a small bag that is incompletely separated from stomach, and it has got the vagal and sympathetic supply. We, for to understand this particular thing, you need to know what vagal supply is, sympathetic supply is, because I already told you how the stomach is innervated, how it is supplied, and what is the nervous backup. That is discussed in detail in the first part. So please go back to part one, see that, and then only see the rest of the discussion. Otherwise, you won't understand anything. So the Pavlov pouch has got vagal as well as sympathetic supply. It is just a separate bag-like structure which is separated from the stomach. And the use is, it is used to demonstrate the cephalic and the, sorry, only the cephalic phase of the uh, gastric secretion, and also the role of vagus nerve in gastric secretion. That is Pavlov pouch. Then we have another pouch known as the Farrell and Ivy pouch. The Farrell and Ivy pouch is the same Pavlov pouch which is transplanted into the subcutaneous tissue. And then it is denervated. That means there is no nerve supply and it has only got blood supply. So, what can be studied here? In the blood, there will be hormones. So, the hormonal regulation of gastric secretion can be studied with the help of Farrell and Ivy procedures or Ivy experimental methods. Then we have sham feeding. Sham feeding is a procedure in which the esophagus is cut in the neck and the food will exit before reaching the stomach. Okay, this was done on dogs. This particular experiment was done on dogs. So the esophagus was cut at one end, then we give food to the dog. So the food has, so sorry, the dog has got a satisfaction that he has already had the food. But however, since the food is coming out, it is exiting before reaching the stomach. Okay, and if there is some secretion that is coming inside the dog's mouth, that means the cephalic phase is activated there. So it is used to study the cephalic phase. After vagotomy, we see that after vagotomy, the secretion stops. There is no secretion. That means the cephalic phase has got some relation to the vagus nerve. Right? So in order to study all this, we have these three experiments. So now let's see what are the phases that I'm talking here, like about what is this cephalic phase, what is the gastric phase, etc. So the first phase of gastric secretion is the cephalic phase. The second phase is the gastric phase. And there is another phase, that is the intestinal phase. And another phase is also present, that is known as the interdigestive phase, which is the fourth phase. So cephalic phase is, it is controlled by conditioned reflex as well as unconditioned reflex. In today's session itself, I told you what conditioned reflex and unconditioned reflex is. In conditioned reflex, even the sight, smell, or thought of the food or hearing about food will trigger salivation. Whereas in unconditioned reflex, the presence of food in the mouth triggered salivation. So similarly, this conditioned reflex as well as conditioned reflex triggers the gastric secretion, because here we are talking about the gastric secretion, not salivary secretion. And then this is controlled by the vagus. So when the vagus is stimulated, there is secretion of gastric juice. So as simple as that. Now moving to the gastric phase, which is under the control of the nervous and the hormonal phase. What happens is this phase is entered when the food reaches the stomach. So the vagus nerve is activated, the local nerves are activated, and also gastric enzyme is also activated. So the stimulation leads to secretion of gastric juice. So in the gastric phase, we have the functioning of vagus nerve, local nerves, as well as gastric. And when the food comes into the intestinal phase, that is when once the chyme or the food bolus enters the intestine, the duodenum, then what happens? The gastrin plays an important role. Then another, so gastrin is a stimulatory enzyme that helps in stimulation or secretion of the gastric juice. But there are certain inhibitory ones like secretins, CCK, that is the short full form is given there, cholecystokinin, pancreazoamine. Then we have also got other structure, other enzymes like GIP, that is gastroinhibitory polypeptide, then VIPs, vasoactive intestinal polypeptides. VIP, I already told you in salivary secretion, right? So that leads to inhibition. So there is no secretion of gastric juice. Okay, so that is the intestinal phase. I hope it's clear. So, and the last one is the interdigestive phase. So, interdigestive, as the name suggests, it is in between the meals or when there is fasting and all, this particular phase comes up. So, it is mostly under the control of gastrin. Okay, so here what happens is the small amount of juice is produced, and let's see what is the percentage of gastric juice produced in these stages. So the total percentage of gastric juice produced in cephalic phase is 30 percentage. The total amount produced in gastric phase is 60 percentage. Then that produced in intestinal phase is 10 percentage, and in interdigestive phase it is very minimal. We can't say how much it is, right? It is very minimal. And I have just compared this particular gastric phase regulation with the pancreatic phase regulation. So, this we will discuss in the next session when we talk about what pancreas is, what are the secretions of pancreas. But just have a quick look. Here, in the cephalic phase, only nervous system was controlling the cephalic phase. Here also, the nervous system is controlling. Then the gastric phase, which is here also it is gastric phase, it is controlled by hormonal only. But here both nervous and hormonal plays a role. And in intestinal phase it is mostly hormonal, and in the intestinal phase here also it is hormonal. Okay, and in cephalic phase of the stomach, sorry, of the gastric secretion, we saw that it was via the vagus nerve. Here also it is via the vagus nerve. And in gastric phase, since it has got both nervous and hormonal, we had the vagus nerve as well as gastrin here. But since it is only hormonal, we have only gastric. There is no vagus nerve in the gastric phase. And when you come to intestinal phase, since it is mostly hormonal, we had mostly the functioning of gastrin. But here in intestinal phase, we had more of secretin and cholecystokinin. So secretin will help in production or secretion of pancreatic juice, which is rich in bicarbonate, and cholecystokinin helps in secretion of pancreatic juice, which is rich in enzymes. Okay, so that is the difference. So just for comparison, I have put this particular thing here, which is very much similar, but only with slight differences. The major difference is in gastric phase of gastric secretion, it is mostly under the control of nervous and hormonal, whereas it is only hormonal when it comes to pancreatic secretion. Okay, we will have a separate discussion on pancreas, so don't worry. Now let's see some important applied physiological problems that is seen. First one is gastritis. What is gastritis? It is the inflammation of the gastric mucosa. So this can be due to various reasons like H. pylori infection. Okay, it can be due to certain NSAIDs, which are the drugs. Okay, drugs means some tablets that we take, anti-inflammatory tablets. Okay, non-steroidal anti-inflammatory drugs. So they can lead to gastritis. Then some type, it can be due to some nasogastric trauma, autoimmune diseases, anorexia, indigestion. Okay, all this. Then there is something known as gastric atrophy. Gastric atrophy. So, gastric atrophy is the shrinkage and weakening of stomach muscles and glands. So, the cause is chronic gastritis, autoimmune diseases. Not AIDS, it is autoimmune diseases. Okay, autoimmune diseases. Then, these are some of the reasons. Then we have peptic ulcer. Peptic ulcer is the erosion of stomach or the duodenal wall. Okay, so we can call it gastric ulcer as well as duodenal ulcer. So if it is occurring in the stomach, it is known as gastric ulcer. If it is occurring on the duodenal wall, it is known as duodenal ulcer. Can be due to excess of pepsin, excess of HCl, reduced mucin, because mucin was highly protective. So when this mucin is reduced, it can also lead to acidity, then leading to peptic ulcer. So the features include a form of burning of the burning epigastric pain, which is during meals in gastric ulcer, which is this is an important thing that I'm saying. Okay, so there is epigastric pain during meals in case of gastric ulcers, whereas there is epigastric pain 1 to 2 hours after meals and at nights in case of duodenal ulcer. Okay, so there will be nausea, vomiting, hematemesis. That means there will be vomiting of blood, then heartburn, heart pain, anorexia, weight loss, everything will be also present. Okay, so this is about the secretion of the gastric enzymes, particularly HCl is perhaps one of the most important topics that can come from this topic. So it is released by the parietal cells or the oxyntic cell, and this diagram should be learned properly and should be drawn for your exam. And we have also seen what are the various enzymes released by the gastric secretion. So in the next session, we'll wind up the gastrointestinal system after learning about the pancreatic secretion, how it is regulated, and the rest of the chapter. See you all. Bye.